Hi everybody, and welcome to our module on transcription. Transcription is the process of synthesizing RNA using DNA as a template. And by way of review, what differentiates RNA from DNA is that RNA contains ribonucleotides, not deoxyribonucleotides, and also RNA contains uridine instead of the nucleotide thymidine. If you look at the bottom of the screen, this is the structure of thymidine on the left and uridine on the right. Note that uridine has an OH group here hanging off the sugar molecule. That makes it a ribonucleotide. Thymidine does not have an OH group here. That makes it a deoxyribonucleotide. To understand the process of transcription, let's take a look at this cartoon I've made of a double strand of DNA. At the top we have a strand of DNA going from 3' to 5', and anti-parallel to that we have a complementary strand of DNA that runs from 5' on the left to 3' on the right. I've denoted all the different nucleotides and bases by colors, and they are complementary to each other so that adenosine is always paired with thymidine and cytidine is always paired with guanosine. The big picture of what's going to happen in transcription is that the two DNA strands will separate, and one of the strands will be used as a template. That's called the template strand, and it will be used as a template to synthesize RNA in the 5' to 3' direction. And in the video on DNA replication, I explain why nucleotides are always added to growing strands of DNA or RNA in the 5' to 3' direction. This process will yield an RNA molecule that is complementary to the template strand in terms of its bases, so that anywhere that the template strand has thymidine, the RNA molecule will have adenosine, and anywhere the template strand has cytidine, the RNA molecule will have guanosine. The only difference is that the nucleotides in the RNA molecule are ribonucleotides, not deoxyribonucleotides, and also the RNA strand has uridine, where the DNA strand may have thymidine. So if you wanted to know what this RNA strand was going to look like, you could look at the template strand and in your mind convert all the bases to the complementary base, and then you would have a picture in your mind of what the RNA will look like that comes from this template strand. However, there's no need to do that, because if you simply look at the other DNA strand at the bottom of the screen, it shows you what the RNA strand will look like that is synthesized from the template strand. This DNA strand on the bottom is complementary to the template strand. Therefore, it is showing you what the RNA from the template strand will ultimately look like. And for that reason, this strand on the bottom is called the coding strand, and this is the strand that you'll often see shown in books and in research papers. It's much easier than showing the template strand, which you have to convert in your mind into mRNA by flipping all the bases. The coding strand instead is often shown, and that tells you what the mRNA will look like that ultimately comes from the template strand. Make sure you understand that difference, and make sure you understand what you're looking at when you see a coding strand illustrated in one of your textbooks. So for example, you might read in one of your textbooks that a piece of DNA has the sequence GTCA, and that via transcription that makes a piece of RNA that has the sequence GUCA. And this might be confusing to you because you don't understand why the bases aren't complementary. And the reason is because this piece of DNA is the coding strand, not the template strand. So when you look at the coding strand, whatever you see for nucleotides is the same thing you will get in terms of bases in the RNA. The difference is that you won't have thymidine in the RNA, and the RNA will be made up of ribonucleotides, not deoxyribonucleotides. So now that we understand that, let's talk about some of the many different kinds of RNA that you can find inside of cells. Messenger RNA is the RNA that codes for amino acids and proteins. These are the longest chains of RNA, and the nucleotides in a strand of messenger RNA specify amino acids that can be used to make proteins via the process of translation. Ribosomal RNA is also very important for translation. Ribosomal RNA forms ribosomes, and ribosomes are the site within cells where proteins are synthesized. Transfer RNA is used to transfer amino acids to proteins. It is also very important for the process of translation. MicroRNA are a special form of RNA that we'll talk about in more detail at the end of this video. They can regulate gene expression. MicroRNA target messenger RNA molecules. They bind via base pairing, and they block the translation into proteins. So just because DNA gets transcribed into mRNA doesn't necessarily mean that mRNA will go on to form proteins. There are other RNAs around which can block the conversion of mRNA into protein, and therefore they regulate the expression of a gene into protein. MicroRNAs are one such type of RNA. Small interfering RNAs are another type of RNA that regulate gene expression. They can cause degradation of messenger RNA so it never goes on to form proteins. And then finally, small nuclear RNAs are a type of RNA that is important for splicing. We'll talk more about this later. RNA polymerase is the enzyme that synthesizes RNA using DNA as a template. It shares some similarities with DNA polymerase, which also synthesizes chains of nucleic acids using DNA as a template. But one very important difference is that RNA polymerase does not require a primer like DNA polymerase. In the video on DNA replication, I talk about how DNA polymerase works, but it requires a short sequence of nucleic acids in order to get started. That's called a primer. RNA polymerase does not require a primer. RNA polymerase works by binding to regions of DNA called promoter regions. It requires proteins called transcription factors to work, and it binds to DNA and opens the double helix and begins the process of transcription so that RNA will be made from DNA. In prokaryotes like bacteria, there is one RNA polymerase enzyme. It is a multi-subunit complex, and it makes all the different types of RNA like messenger RNA, tRNA, and ribosomal RNA. In eukaryotes, there are multiple different RNA polymerase enzymes that function in cells. RNA polymerase 1 makes most of the ribosomal RNA. Ribosomal RNA comes in a number of different sizes, and RNA polymerase 1 makes the 5.8S size, the 18S size, and the 28S size. RNA polymerase 2 is the RNA polymerase we're going to talk a lot about in this video. It's used to synthesize messenger RNA. And then RNA polymerase 3 makes some forms of ribosomal RNA. It makes the 5S size, and it makes some of the other forms of RNA found in cells. There are some naturally occurring substances and also some drugs that function by inhibiting RNA polymerase. One of them is alpha-amanitin. This is a protein produced by the mushroom Amanita phalloides, which is also called the death cap mushroom. This protein is a powerful inhibitor of RNA polymerase 2, and if you consume this mushroom, the protein will be taken up by your liver cells, and this will lead to liver failure. And unfortunately, I've seen one person in my career who accidentally ate one of these mushrooms. She ended up in the intensive care unit with acute liver failure and required a liver transplant. It is very dangerous to eat these mushrooms, hence the name death cap mushrooms. Rifampin is a drug that inhibits the bacterial version of RNA polymerase. It can be used in the treatment of tuberculosis infections. And actinomycin D is a chemotherapy agent. It inhibits RNA polymerase and therefore blunts the replication of cancer cells. Back to our discussion of transcription, I mentioned before that transcription factors are proteins that are required by RNA polymerase in order to initiate transcription. In prokaryotes or bacteria, there is a single transcription factor called protein factor or sigma factor. Eukaryotes have multiple different transcription factors. Many of these bind to RNA polymerase 2, the RNA polymerase used to synthesize messenger RNA, and they have names that begin with TF for transcription factor like TF2D, TF2B, and TF2E. Promoters are DNA regions that are not transcribed, but they can bind to RNA polymerase as well as transcription factors, and therefore they promote the process of transcription. And once RNA polymerase has bound to a promoter region, it can open the double helix and begin the process of synthesizing RNA. And promoters are generally located just before a gene that's going to be transcribed, as I've shown at the bottom of the screen. If this is the transcription start point, the promoter will be just before that. Following the start point will be the gene that needs to be expressed. There are a number of promoters that have been described. The Tata box is one of the famous ones. It's a very common eukaryotic promoter. It gets its name because the nucleotides are TATAAA, and it can bind to transcription factors, especially TF2D. The cat box is another promoter region that's been described. It gets its name because the sequence of nucleotides is CCAAT, and then there's also a GC box, which has been described because it has G and C nucleotides. Besides the promoter region of DNA and the gene to be expressed, there are other portions of DNA that are important in determining the rate of transcription of a particular gene. One of these regions of DNA is a region called an enhancer. Enhancers are DNA sequences that increase the rate of transcription. They can be found either upstream or downstream from the gene that they regulate, and they bind to special transcription factors called activators. And once they have bound to the activator, they stabilize transcription factors and RNA polymerase, and this leads to more transcription of a gene. Now, one thing that's interesting about enhancers, and one thing you should know for step 1 of your boards, is that because of DNA coiling, the enhancer region of DNA may be geometrically very close to the gene that is being transcribed, but may be many, many nucleotides away. For example, if this is our gene to be transcribed, we may have an enhancer region that is way upstream, but because of DNA coiling, it's twisted around and it ends up being geometrically very close, and therefore it can function as an enhancer for a gene that's very far away in terms of the number of nucleotides. And silencers are just the opposite of enhancers. They are DNA sequences that decrease the rate of transcription. They can also be upstream or downstream from the gene they regulate. They can also be very far away in terms of nucleotides, but geometrically close. These silencers bind to transcription factors called repressors, and when they do so, they can prevent RNA polymerase binding and therefore prevent transcription. Untranslated regions, as the name implies, are portions of an mRNA molecule found at the 5' and 3' ends that are not translated into a protein. At the 5' end, there's an untranslated region that is upstream from the coding sequence, as I've shown at the bottom of the screen. That's this untranslated region here on the left. And this untranslated region is important because it's recognized by ribosomes to initiate the process of translation. There's also an untranslated region at the 3' end. This is always found following a stop codon, and this untranslated region is important for post-transcriptional gene expression. So just because this molecule of mRNA has been transcribed from DNA, it doesn't necessarily mean it's going to go on to make a protein. This untranslated region here has a lot of influence over that because it can bind to other factors that can determine whether this mRNA becomes translated into a protein or not. So in bacteria or prokaryotes, the process of protein synthesis is relatively simple and straightforward. One of the reasons for this is because there is no nucleus, so DNA is free-floating in the cytoplasm. It is transcribed into mRNA, and then the mRNA is translated into proteins. In eukaryotic cells, the process of protein synthesis is more complicated. First of all, DNA is found inside the nucleus, and in addition, it contains alternating regions called introns and exons. The exons will go on to be translated into proteins, but the introns will be cut out. The way this works is that the DNA is transcribed into RNA containing the introns and the exons. Then the introns are cut out inside the nucleus, and then the RNA exits the nucleus, and out in the cytoplasm is where the ribosomes can be found, and they are used to translate the RNA into protein. So I've summarized these points at the top right. Introns stay in the nucleus, exons exit the nucleus, and go on to be translated into protein. And cutting out the introns is only one of several modifications that occur to mRNA in eukaryotes. The initial transcript of mRNA from DNA in eukaryotes is called hnRNA, which stands for heterogeneous nuclear RNA. It's also sometimes called pre-mRNA. This version of mRNA becomes modified extensively to become the mRNA that exits the nucleus, goes into the cytoplasm, and is ultimately translated into protein. And there are three key modifications that occur to RNA before it leaves the nucleus, and you need to know about all of these for step one. The first one is called 5' capping. The second one I briefly mentioned on the last slide, that is splicing out of introns. And the third one is called 3' polyadenylation. And we'll talk about all of these in the next few slides. 5' capping is the addition of a special nucleotide called 7-methylguanosine to the 5' end of RNA. This is added to RNA soon after transcription begins, and it distinguishes mRNA from other forms of RNA. Basically, this 5' cap is placed on RNA to tell the other organelles in the cell that this is a piece of mRNA. If you look at this cartoon I've shown on the screen here, we have a molecule of mRNA going in this direction toward the 5' end. Remember, the 5' end always has the phosphate groups like shown here. At this 5' end, a molecule of 7-methylguanosine has been added. This is a ribonucleotide just like the guanosine you would find in RNA, except that it has a methyl group hanging off the 7 position. Hence it is called 7-methylguanosine, and this caps the 5' end of RNA to tell other structures in the cell that this is a piece of mRNA. RNA splicing is the second important modification that happens to mRNA in eukaryotic cells. RNA splicing occurs during transcription. This is the process by which introns are removed from mRNA. It occurs in the nucleus of eukaryotic cells. And introns can be identified because they always have two specific nucleotides at either end in an mRNA molecule. If you look at this diagram at the bottom of the screen, this is the intron in dark blue in the middle. At the 5' end, there's always a GU to denote the beginning of the intron, and at the 3' end, there's always an AG. There's a complex process by which RNA splicing occurs, and they like you to know something about this for the step 1 exam. So the primary transcript, the initial piece of mRNA, combines with structures called snRNPs in order to undergo RNA splicing. snRNPs are small nuclear ribonucleoproteins. They are short RNA polymers complexed with proteins, so they're a mixture of RNA and proteins. And the RNA in these snRNPs has a high content of uridine, so they're sometimes referred to as uRNAs. You will read in your textbook that snRNPs have lots of uRNA. And there are 5 different subtypes of uRNAs that have been identified: U1, 2, U4, U5, and U6. When the snRNPs and the mRNA bind together, the entire structure is called a spliceosome. At this point, a loop of mRNA containing the intron will be formed. This loop is called a lariat. The lariat will then be released, and that takes the intron away with it, and then the exons are joined. And here's a cartoon from the web just to help you better remember how this entire process of RNA splicing works. So this is our intron here between 2 exons. It's marked by a GU at one end and an AG at another end. The lariat forms here by releasing the GU end, and then the lariat is removed and the 2 exons are brought together, and the lariat is left to go away free and take the intron with it. In some connective tissue disorders, antibodies form against elements of the spliceosome, and I just want to briefly review those here. In the immunology section in the video on lupus, I talk about anti-Smith antibodies. These are antibodies against proteins in the snRNPs, and you can see elevated levels of anti-Smith antibodies in patients who have lupus. Anti-RNP antibodies are antibodies against proteins associated with the U1 RNA, and these antibodies are strongly associated with a rare autoimmune condition called mixed connective tissue disease. If you ever hear positive U1 RNAs, you should think of this very uncommon disease because these antibodies are so strongly associated with it. You can also see elevated levels of anti-RNP or anti-U1 RNA antibodies sometimes in lupus or scleroderma. You might wonder why eukaryotic cells have all these introns and exons, and the reason is because it allows many different proteins to be made from the same gene. For example, if you have a piece of DNA in a gene that has exons 1 through 10, you could synthesize one protein by combining exons 1, 3, and 7, and a second protein by combining exons 2, 5, and 10. Thus, from the same gene, the same genetic material, you have made 2 different proteins, and this is what allows eukaryotic cells to be more advanced and high functioning than prokaryotes. And here's a cartoon from the web just to make this even more clear. We have a gene, a piece of DNA with 5 exons, each denoted by a different color. That is used to transcribe a piece of messenger RNA that also contains the 5 exons. Via alternative splicing, however, we can make protein A by combining all 5 exons. We can make protein B by combining exons 1, 2, 4, and 5. And we can make protein C by combining exons 1, 2, 3, and 5. So as you can see, one gene, one piece of DNA used to make one piece of messenger RNA, but then alternative splicing used to generate multiple different proteins. Splicing errors have been described in some disease states. A mutation can lead to a loss of an exon, a retention of an intron that should be cut out, or incorrect joining of introns instead of cutting them out. In beta thalassemia, there are a number of genetic mutations that have been described, and some of them involve the splice sites. And in certain cancers, many splice site mutations and other errors have been described. The final modification that occurs in eukaryotic mRNA is called 3' polyadenylation. This modification occurs at the termination of mRNA transcription. In other words, it happens when the cell is done transcribing the gene into mRNA. The trigger for this to occur is a specific sequence of DNA and RNA nucleotides. If you look at the bottom of the screen, I've showed the coding strand of a piece of DNA. This will code for a piece of mRNA that has an AAU-AAA sequence. This sequence is called the polyadenylation signal because it's the signal for the gene to stop transcribing and to add a series of adenosine nucleotides called a poly-A tail to the piece of mRNA. So what this means is at the end of a gene that is being transcribed, you will usually find AAU-AAA, the polyadenylation sequence, followed by 10 to 30 nucleotides and then a CA. And I've shown this at the bottom of the screen here to help you remember. When this sequence shown at the bottom of the screen is encountered in a piece of RNA, that triggers the process of 3' polyadenylation. This process requires several RNA binding proteins. One of them is called CSF, which stands for cleavage and polyadenylation specificity factor. This is what binds to the polyadenylation sequence AAU-AAA. Another important RNA binding protein is called CSTF, or cleavage stimulation factor. This binds to the CA sequence that is also always found at the end of genes. And binding of these 2 factors leads to termination of DNA transcription. Once DNA transcription has ceased, the enzyme poly-A polymerase comes in and adds about 200 adenosine nucleotides to the 3' end of mRNA. This is called a poly-A tail, and this process is called 3' polyadenylation. Poly-A polymerase uses no template. It just pulls adenosine nucleotides and adds them to the end of the mRNA molecule. It also lops off a bit of the mRNA molecule, and what you're left with is what I have shown down at the bottom of the screen. You've got that polyadenylation signal followed by a short stretch of nucleotides followed by a poly-A tail at the 3' end of the mRNA molecule. So now that we've gone through all the elements of transcription, let's go over this summary slide and it should all make sense to you. So at the top of the screen, I've shown a DNA coding strand. We have the 5' end on the left side of the screen, the 3' end on the right. We start with the promoter region, which has the cat box and the tata box, followed by the transcription start point. We then have a series of exons and introns which end in the polyadenylation signal that will signal the end of transcription. This coding strand is transcribed into a piece of mRNA that goes through all the modifications we just discussed. So it's got the cap at the 5' end, it's got the poly-A tail at the 3' end, and all the introns have been spliced out to form a piece of mRNA. That can now go to the cytoplasm to begin the process of translation into a protein. Now, just because a gene has been transcribed into messenger RNA, that does not necessarily mean that piece of messenger RNA will be translated into a protein. There are a number of elements within cells that can influence whether or not a piece of messenger RNA is translated into a protein. One of those elements is microRNA, which I mentioned at the beginning of this video. MicroRNA molecules are important regulatory molecules for messenger RNA. MicroRNA can regulate messenger RNA expression into proteins. The way microRNA does this is by binding to mRNA via base pairing. If there is extensive binding between a piece of microRNA and a piece of messenger RNA, this can remove the poly-A tail. When this is done, this exposes the messenger RNA to degradation by endonucleases in cells. So what this means is that microRNA is a way of modifying gene expression at the mRNA level. The DNA has been transcribed into mRNA, but because of microRNA, the mRNA does not go on to translate into a protein. Other elements within cells that can influence whether a piece of mRNA is translated into a protein are the processing bodies or P-bodies. Some messenger RNA is moved to P-bodies in cytoplasm. These are organelles within cells. This has been described as occurring in pieces of messenger RNA that bind microRNA but to a less extensive degree. The mRNA then is sequestered in the P-bodies from ribosomes so it can't be translated. Often the messenger RNA is degraded, although there is also some evidence that the messenger RNA may be sequestered and then later released to be translated into proteins. But the point here is that the P-bodies are another mechanism by which a gene that has been transcribed into mRNA can be affected so that it does not necessarily go on to be translated into a protein. And that concludes our module on transcription.